There's a bug on your logo.
So right now we're not studying bugs. We're studying life science. Well, bugs are kind of life science too, but so we're going over my notes for cell biology in a nutshell. So scientists obtained the first structural snapshot of an entire ribosome, which is an important organelle in charge of protein synthesis, in 1999. This more recent image captures a bacterial ribosome in the act of making a protein, the long straight spiral. Um, in the lightest shade of blue in this image right here, it shows that unlike typical cellular machines, which are clusters of proteins shown here as purple ribbons, ribosomes are composed mostly of RNA, the large light blue and gray loopy ladders. So, detailed studies of ribosomal structures lead to improved antibiotic medications because they create different kinds of proteins and enzymes. The ribosomes do. So, so here in this molecule, it's a little bit difficult to tell the colors, but you have the RNA is the most prominent part of the ribosomes. The blue and the gray are the RNA subunits, which are created in the nucleolus inside the nucleus. This long straight strand that looks like a helix is a protein being synthesized. So in the very light blue, okay, and the purple ribbons that are interspersed with the RNA, large RNA subunits, are proteins as well that help with the function of the ribosome as well. So the next thing we're gonna learn is about an organelle called peroxisomes. These are tiny structures that are in charge of detoxifying alcohol, um, and they use oxygen to break down fats as well. It They contain reducing enzyme catalase. They usually have also some oxidases. Microbodies are roughly so they're microbodies, usually roughly spherical in shape. They're bound by a single membrane. Usually they are zero point 0.5 to 1 micrometer in diameter. There are several types, by far the most common of which is the peroxisome. Peroxisomes derive their name from hydrogen peroxide, a reactive intermediate in the process of molecular breakdown that occurs in the microbody. Peroxisomes contain type 2 oxidases, which are enzymes that use molecular oxygen in reactions to oxidize organic molecules. A product of these reactions is hydrogen peroxide, which is further metabolized into water and oxygen by the enzyme catalase. Catalase is a prominent constituent of peroxisomes. Also, peroxisomes contain other enzyme systems that degrade various lipids. The plant glioxysome is a peroxisome that also contains the enzymes of the glyoxylate cycle, which is crucial to the conversion of fat into carbohydrates. There are many ways that peroxisomes are similar to lysosomes. They are small vesicles, so we learned about lysosomes earlier, which are also in charge of breaking down macro or large molecules. So they're both small vesicles found around the cell. They both have a single membrane containing digestive enzymes for breaking down toxic materials in the cell. However, peroxisomes different that they have different types of enzymes that they hold from lysosomes. Peroxisomes hold enzymes requiring oxygen, or which are called oxidative enzymes. Lysosomes have enzymes that work in oxygen poor areas as well, and at lower pH. Peroxisomes absorb nutrients that the cell has acquired. They are very well known for digesting fatty acids. They play a part in the way organisms digest ethanol, which is an alcohol. They they liver cells are expected to have more peroxisomes than most other cells in the human body for the reason of um, them needing to break down toxic molecules or substances. So they also play a role in cholesterol synthesis and the digestion of amino acids. Their enzymes attack complex molecules and break them down into smaller molecules. One of the byproducts of digestion of peroxisomes is H2O2, which is hydrogen peroxide. Peroxisomes have developed to a point where they're able to contain the hydrogen peroxide, break it down into water, H2O, and oxygen, O2. So the water is harmless, and oxygen is used in various reactions and in the subsequent digestive reactions. In summary, peroxisomes contain enzymes that oxidize certain molecules normally found in the cell, notably fatty acids and amino acids. Those oxidation reactions produce hydrogen peroxide, which is the basis of the name peroxisome. Peroxisome breaks down very long chains fatty acids through beta oxidation. In animal cells, the long fatty acids are converted to medium chain fatty acids, which are subsequently shuttled to mitochondria, where they are eventually broken down to carbon dioxide and water. So the fatty acids are turned into carbon dioxide, CO2, and water. Peroxisomes are thought to acquire their proteins by selective import from the cytosol. Peroxisomes resemble the endoplasmic reticulum in being a self-replicating membrane-enclosed organelle. Peroxisomes have a single membrane surrounding the digestive enzymes, and also dangerous byproducts of their work like hydrogen peroxide. The protein enzymes are usually created by the lysosomes floating in the cell, which insert proteins into the peroxisome bubble. Um, so the lysosome contributes um, these enzymes as well. Thank you.